Hey guys, it's Mr. Kennedy, and I'm back to talk a little bit about transcription. And remember, transcription is in the entire process of going from a gene to a protein. Now, oftentimes you'll hear this process, protein synthesis, called the central dogma, and here it is down here at the bottom. It goes from DNA replication, from DNA to RNA, which is transcription, and from RNA to protein, which is translation to a trait. We're going to do the first part, okay? Now, transcription is, as I said, is basically turning DNA, DNA to RNA. This is transcription. Now, a few things we need to know. RNA is a little bit different than DNA in three ways. It has ribose sugar where DNA had deoxyribose. It has the nitrogen bases A, U, C, and G, so it has U instead of thymine. It's single-stranded instead of double-stranded. And there are a lot of different types of RNA that we're going to talk about: mRNA, tRNA, rRNA. All right. Now, transcription is making mRNA. Messenger RNA is what transcription is. Now, what it does is it transcribes a DNA strand. When a DNA splits apart, this is something you didn't know necessarily in biology. There's two strands. There's a template strand and the untranscribed, the coding strand. You can see down here at the bottom. Okay, you can see down here at the bottom. Here's the coding strand, the strand here that's not being turned into mRNA. And here's the template strand, the one that is being turned into mRNA. So you have the template strand and the coding strand. The coding strand is going to be the same as the mRNA, except for where there's T's in the DNA, it'll be U's in the RNA. All right, and last thing is there is an enzyme that does this, just like they did DNA replication and everything else. The enzyme is RNA polymerase, which is in charge of transcription. You need to remember that. All right, so if we look at RNA polymerase, there are actually three. There's RNA polymerase 1, 2, and 3. I know, ingeniously named. But RNA polymerase 1 transcribes rRNA, which is ribosomal RNA. RNA polymerase 2, which is the one we're concerned about most often, transcribes mRNA. And then RNA polymerase 3 transcribes tRNA, which is a transfer RNA, which we'll talk about later on in protein synthesis. Now, when we talk about how a gene is actually read, there are two regions. There's a promoter region and the enhancer region. The promoter region is the binding site before the gene, and it has what's called a ta-ta box. And the reason that it's called a ta-ta box is because it always starts with the exact same sequence: TA, TA, AA, A. And that is called the ta-ta box. And this is the binding spot for RNA polymerase. So this is where RNA polymerase starts. And then you have the enhancer region. This is the binding site far upstream, and this turns transcription on, because transcription doesn't actually take place until it reaches the template of DNA where it needs to transcribe. Now, there are some transcription factors that help bind the RNA polymerase to the promoter region, and they can either turn on or turn off transcription, and it varies and it varies. The next thing is you have to match the RNA basis to the DNA basis. So if we're looking down here, how it would go is this: wherever you have a C, you're going to have a G. A T is going to be an A will be a U because it's an RNA. A G will be a C will be a G, etc. So it'll match it just like you did whenever you dealt with DNA replication, except for U's or T's. Now, transcription really happens in three processes. The initiation, which is when the ta-ta box comes into play and RNA polymerase starts the initial transcription. Elongation is whenever you have RNA polymerase unwind the DNA and it starts adding nucleotides to the 3' end, just like it did in DNA replication. And then you have termination, when the RNA polymerase actually releases and stops the sequence. Now, in eukaryotic genes, we have a lot of junk DNA. So we have a lot of our DNA that codes for nothing. Well, basically the DNA is broken up into two regions. It's broken up into exons, which are the real genes, and the introns, which are the junk, the in-between spaces. And you can see down here on the diagram, it's going to vary from place to place. So we only want to code for certain things. Now, since we only want to code for a certain thing, we're going to have what's called an mRNA splicing, because we only want the real gene or the real copy of the DNA, right, that's actually going to be useful. So what happens is we have pre-mRNA, which is made with both the exons and introns attached. Then you have mRNA splicing comes in, which takes out the introns, and that creates the mature RNA. All right, there's a couple of RNA splicing enzymes. There's the SNRP, not to be confused with the Smurfs, but the SNRPs are small nuclear RNAs. They're proteins, and they help to take out the introns and connect the exons together. And then they have the spliceosomes, which are when you have several SNRPs together, and they cut and paste the gene to get what you want to actually occur. Remember that the entire process of transcription is taking place in the nucleus because DNA can't leave. So we need to protect the mRNA as it leaves the nucleus and goes to the cytoplasm, to the ribosome. And the way we do this, there's enzymes in the cytoplasm that will attack mRNA. So we protect the ends of the molecule by adding a 5' GTP cap to one end and a poly A tail to the other. And this will help protect the mRNA from these enzymes that may attack it when it leaves the nucleus. All right, I hope this is a quick little overview of transcription. I hope it helps.